So today we are going to speak or learn about molecular markers. You have already studied the RFLP, AFLP as well as RAPD uh, animation techniques. Now I'll be giving you the overall idea and the depth of the topic, what exactly is these techniques are. So basically molecular markers are nothing but a sequence of a DNA, and this sequence of the DNA which will be used to detect certain inheritance patterns or to understand what exactly is the variations of the genes between the different individuals as well as the certain polymorphism characteristics. So what is it? We will understand clearly. So yes, so just imagine you have an entire genome sequence of your human or let's say any of a bacterial strain. Now, you do not know everything about this DNA, but then let's say you have a marker DNA. What is a marker DNA is nothing but a certain oligonucleotides, that is nothing but your nucleotides, which is able to complementarily bind to your part of the DNA sequence of that entire genome. You will be able to have a forward and reverse kind of a primer. You will be able to amplify that entire DNA. So the molecular markers are nothing but a certain markers which is able to bind to certain DNA or certain amplify the DNAs. Also, they'll be able to understand the difference between a normal human and the person who is having certain genetical disorders, all those things. So when we speak about the marker system, we have two different systems of the marker systems. One is a morphological marker and other one is a molecular marker. The morphological marker is what exactly we have learned in the Mendel system. You are able to see the changes through the phenotypic variations. Uh, Basically, if you are able to have a same variety of a plant, but one plant is tall and the other one is a dwarf plants, or particularly let's say uh, you have flower size and how exactly the petals are kept, how exactly the colors of the flowers are, the certain phenotypic variation, the differences you are able to see. Under molecular markers, again we characterize into a biochemical marker and genetic marker. Now, biochemical marker is nothing, we have replication, transcription, and translation, right? So, we know our part of a DNA gets transcripted, and from that transcription, we have a translation. Now this translation is nothing but a proteins. Now these proteins, whether I and someone who is deceased having a same kind of a proteins. Now let's say a person is a hemophilia affected. Probably his proteins which are expressed are different from the person who is not hemophilic or a healthy person, right? So we call it as a biochemical marker specifically used for the protein. So person who is not having a, let's say, a certain enzyme production, the one more person is having a healthy enzyme production, so the changes will be there in the fragments of these proteins, and these are called biochemical markers. Next comes the genetic markers. The genetic markers are usually nothing but the DNA markers. So we have the different techniques to understand what is the difference of the DNA between two individuals or between the strains, between the two strains of the bacteria or within some certain species, all those things. Now, various techniques are used to understand those. Those are RFLP technique, AFLP technique, and RAPD technique. Now, in these techniques, we are mainly depending on the sequence of a DNA. So under this technique, the RFLP is called non-PCR technique. The reason is we are not using PCR method, that is polymerase chain reaction method, to understand the to technique. Not this technique is not used for this polymorphism technique. The second one is the AFLP and RPD. The AFLP and RAPD are the PCR techniques. These have the PCR involved with the technique. Now see the DNA markers, we have non-PCR based and PCR based. Non-PCR based is a RFLP technique. So RFLP is nothing but restriction fragment length polymorphism. Now, under PCR based, we have RAPD, AFLP, SCAR, STS, EST, SNP, SSR, and CAPS. These are the various techniques used. Now, we will be particularly concentrating on RAPD and AFLP technique. These are the basic PCR based molecular markers techniques. So, what is an ideal genetic marker? So, what exactly is an ideal genetic marker? So, first thing, it has to be a polymorphic. Polymorphic in the sense it has to have a variation. So, it is able to bind to the different species, let's say different strains. It has to have the uh, yeah uh, variability so that it will be able to bind to the maximum species or strains. So now the codominant inheritance. So what is a codominance inheritance is we have an inheritance like dominant inheritance, let's say, uh, which is of two types, homozygous and heterozygous. So which you will be able to see. Now let's say, are you able to in particularly say this kind of a gene is a homozygous or heterozygous? So now if you are able to understand this heterozygous and homozygous with this molecular marker, so that is what exactly gives you the clarity between them. Otherwise, you will be just would be taking it as a dominant trait or a homozygous trait, or even consider heterozygous trait as an homozygous trait. There will be a confusion. So that variations also should be understandable. So this marker should be like, let's say your entire genome sequence. Now this marker is able to bind to the different spaces. That means you'll be having a quicker amplification. So it should be present or distributed throughout the genome. It should be easy, and the reproducibility, that means even if this marker is you're running in again and again in the different times and the different types of strains, and you are repeating this technique again and again, you should be having the same kind of an outcome. You cannot have a variations with that. So yes, now first we will be going for the RFLP. Now RFLP is restriction fragment length polymorphism. Okay. Now, we all know restriction endonucleases, or we call it as a restriction enzymes. We can use those enzymes to cleave the certain genomic DNA among the individuals of the species. So, we call it as a RFLP. So, in the RFLP technique, before that, see now, this is my restriction enzyme, that is ECOR 1 enzyme. Now, if you see this restriction enzyme, it is able to recognize certain sequence, and after recognizing certain sequence, it is able to cleave the sequence. Now here if you see, it is in the 5 prime end, 3 prime end, it is able to say AATT, recognize that sequence. And then second, if again it is able to palindromically identify TTAA. So it cuts that ends, and it is able to give you one overhang. If you see, a 3 prime end has a 5 prime overhang. Now that doesn't have a double strands, it has a single strand, so we call it as a sticky ends. Now why the sticky ends are important? Because now these sticky ends, just imagine, you know, a particular restriction enzyme is able to cut particular site, and it will give exactly that sequence as a overhang. You can design your primers based on that overhangs. You can design the oligonucleotides which is able to bind to these strands complementary, just because you know what is the overhangs. Now you know wherever the ECOR 1 enzyme is used, you can simply use TTAA as a complementary stance for the overhangs, and then you start your amplification if it is utilized. So this becomes a very easier to understand which primer to be designed for these kind of sequences. So if you take here, we have a different kinds of restriction endonucleases and their restriction site or recognition site. Now, if you see, there are two types of cuts. One we call it as a staggered cut, and one is a blunt end cut. Now, staggered cut is a one which I previously spoke, which gives the sticky ends. That means it gives the overhangs. That is a single stranded overhangs, which is a you see in BAM H1, it is a 3 prime overhang. You can see here, and in the E core 1 it is again a 3 prime overhang, Yes, so all those things. And the blunt end, so you can see the last two enzymes which has the blunt end. Blunt end is it basically does not give the overhangs, it will give a double stranded a proper cut. It does not give a overhangs. So this is called blunt ends. So we have two types of cuttings, one with ends with the sticky ends and one with the blunt ends. Now, sticky ends kind of overhangs we usually prefer because it is easier to design the sequences, complementary sequences for them. With the blunt end, it will be difficult to design the sequences, but usually we prefer the sticky ends for the ligation concepts. Now, how exactly the restriction enzyme works for the RFMP? Okay, this is very important. Why and how it works. Now, just imagine you isolated a bacteria from a soil and you isolated a bacteria from the water, and it turned out to be a E. coli bacteria in the both the scenarios. Okay, now one which you isolated from the soil, we consider it as a strain A, and the one which you isolated from the water, we will consider it as a strain B. Basically, these are the same E. coli bacteria, but they might vary in their DNA sequences, right? So we do not know. So what I do is for these bacteria, I will simply isolate the DNA and simply add my enzyme. I'll be adding one enzyme. We know this enzyme will cut exactly at particular recognition site. If they have the same kind of a DNA, that means they should have the same kind of a cuts and the same kind of a fragments. Now, if you see here in the strain A and strain B, in the strain A, the cuts are such that there is only the fragment 1 and fragment 2 is produced, but the fragment 1 is having just 5 base pairs, and in the strain B, fragment 1 is having approximately 7 base pairs. And the fragment 2 again has a different number of base pairs, and the fragment 2 in the strain B is again having a different kind of a base pair. So that means these might be E. coli bacteria, but they are differ from their genetic sequences. So that is what we say. Even though the bacteria belong to the same E. coli range, they are different strains. That means the genetic makeup is not exactly similar and they are quite different. There now just imagine instead of the bacteria, I have the genetic makeup of a strain A. Simply I'll take a healthy human, and the strain B I will take a hemophilic human. So basically, the healthy human and the hemophilic human have a different genomic sequences. Probably the hemophilic human will have a different DNA, or the inserted or mutated DNA, let's say. So they will also have a different fragments produced. So this is where exactly I'll be identify where exactly they are differing in their DNA sequences, which fragments are different, which fragments they are differing from each other. So that is what exactly we are able to understand in the restriction digestion. Under restriction digestion, so how exactly the entire technique goes. The first you will be having, let's say you have the three tubes, the each one will be isolated the DNA from the three different strains, let's say the E. coli, and then after isolating the three different strain, you will be adding a particular restriction enzyme to this append of tubes, which is with the DNA. Okay, so now this will be having the restriction digestion. During the restriction digestion, once it is done, and then I'll be making it ready for the agarose gel electrophoresis. And then you run the agarose gel electrophoresis. We know the agarose gel DNA being negative, it moves from the cathode, which is negative end to the positive end, which is anode. So then you have the DNA separation based on their sizes. Okay, so the lowest one will have the highest number, I mean fastest one will have the at the lower end, and they will move towards the end, and the lower one, uh, higher molecular weight one, moves little slower. So this is how exactly it goes. And then you transfer it to the southern blotting. It is a blotting technique. So what exactly you're doing is whatever you are having in the agarose gel, exactly copy. You are transferring the exactly DNA onto a nitrocellulose membrane. We call it as the southern blotting technique. So once this is done, the southern blotting technique is done. So This now the membrane is made to complementary bind with the DNA of your interest. Now let's say you know certain sequence of a DNA and you want to find whether this DNA is there in this bacteria. So you will just add that DNA with the radio labeled. That means you will be adding a radio labeled DNA and make them to interact with the single stranded the ends of the DNA here. Uh, Restriction digestion, the ends we have, right? That DNA. So if that DNA binds and then they give the color, fluorescence color over the time, and then you will be able to understand which one is of your interest, as well as you are able to understand what is the difference between these fragments. So this is exactly RFLP technique. So RFLP technique throughout we are not using PCR technique. So next if we see what exactly the advantages of RFLP technique. Simple it is to use, it is non-dependent on the PCR and the codominant markers are utilized. Also disadvantage is you need very high purified DNA. You should be having expertise in the autoradiography. It's a very time consuming and then it's a lot of you need to work on your probes, that is uh, single stranded DNAs with or probes which are able to bind to the sequences which is radio labeled. So that is a drawback. uh, this is used in the measures of the genetic diversity. This is used in the fingerprinting technique. The fingerprinting technique is very easy here. Now let's say you have a DNA sample from the crime scene and you have a suspect. You have three suspects. Now one of them is a criminal. Let's say uh, the digestion which has happened in the crime spot and the criminal should be the similar. So you have the same patterns produced in the gel, and that patterns exactly gives you an understanding what exactly is your who is the criminal. The suspect can be identified. Uh, to understand certain genotypic selections, that is to understand between the two parents and the F1 generation and all can be understood. And then certain marker assisted selections and all those things could be done with the RFLP technique. The second technique is a RAPD technique. It is called random amplified polymorphic DNA. Now you need to understand this is a one technique. You need not know anything about your DNA. You do not need any prior knowledge in a DNA, and you need not know anything about your primer much. Okay, in RFLP technique, at least when you are using your enzyme, you should know that enzyme restriction sites are there in your genomic DNA. That means that enzyme is able to cut your genomic DNA. That kind of restriction sites are available in your genomic DNA. Where exactly in the RAPD technique, you can be absolutely having no clue about your genomic DNA. You just have to isolate your DNA and then you do this RAPD technique. That is why it is called random amplified polymorphic DNA. So basically what are you doing is you are using a DNA sequence. Now let's say you are using from the two different strain again, but here you are using a primer. I know what is a, I hope you know what is a primer. Primer is a short oligonucleotide sequences. Now these primers are basically going to bind to your whole genome randomly at certain places. Okay, and then you are going to amplify the DNA. And here your entire DNA won't be amplified. A part of your amplified DNA will be amplified. So through this patterns, you will be able to understand what is the variations, what is the uh, similarity. So monomorphism and polymorphism we say, which is a similar DNA sequences or fragments are produced, and which are the dissimilar DNA mark, fragments are produced. See how exactly this goes is. Now let's say you have variety 1 and variety 2. Now see in the variety 1, I am using the primer, the double stranded DNA in the black, the primer is in the red. Now in the variety 1, the primer is able to bind to the two sides, one is a forward and the backward. Uh, in the 5 prime, it's a forward, let's say 3 prime is a backward. Sorry, 3 prime is a forward and the 5 prime is a backward. So same way in another spot in the variety, also 3 prime is a forward, 5 prime is a backward. Uh, the two places there is a both the forward and backward primers are able to bind. In the second scenario, only at one place the forward and backward primers are able to bind. The other place it is not able to bind. So now basically, now let's think these are belonging to one of the healthy person. The second one is an unhealthy person. So this is what exactly is the difference between them. So one place it is able to bind, the other place it is not able to bind. So this is the variation. Now just think of it when exactly this will not give the results. So one is you see the circle point. One is it needs to have the forward and backward binding of the primer. Second one is it has to have a minimum distance between the forward and backward binding primers. You can see here first, second and third scenario. The three circles are there. In the first scenario, forward and backward primers are bound and they are in the nearest proximity. Second scenario, second circle, there also they are bound, but they are again in the short distance. In the third scenario, if you see, the forward backward primers are bound, but they are very far away from each other. So now here to bring the amplification is difficult. So if the distance between your forward and backward primer is more than 2000 base spaces, yeah, the application becomes difficult or application does not take place. So this is how exactly you are able to amplify the DNA. Now, what exactly is required in your protocol? So, you just need a bacteria strains. For that strains, you need to have your buffer, magnesium chloride primer, and then you have your DNTP mixed stack polymerase, distilled water, that is molecular grade water, and then you will be uh, so this will be adding to entire of this master mixture. You will be adding to your 25 5 microliter of your DNA, and then one of them you are running without a primer, which is so you are running this and you see this first, second and third. So you will be having three different let's say bacteria, E. coli bacteria only you can see. The one you will be having a larger DNA and the other one will be having a shorter DNA fragments. So you can see one might be representing the homozygote where the other one is representing the heterozygote. So this you can see here in the actual RAPD technique. So this is let's say a particular plant uh, stromata uh, DNA is isolated and then you have the RAPD technique done. So M represents the control and then when you run this uh, wells in the DNA is run. So what happens is certain sequences are similar, fragments are similar, certain are not similar. The not similar one is the dissimilarity or variations within those plants. Where exactly the similar one, exactly they have the similarity. So this is how exactly you can identify the mutations within the same strains or any of the genetic variations like insertion, deletion, or any of the chromosomal aberrations and all those things you can understand with the RAPD technique. RAPD technique, the one of the best thing about RAPD is it need very small amount of DNA. Then like your RFLP which requires a radio labeled DNA, your RAPD technique does not need a radio labeled DNA probes, then does not require probe DNAs also, and then it will be easier to screen here. Here screening of polymorphism is quite higher than the RFLP. Disadvantage is sometimes the inherited dominant traits, you will not be able to understand homozygotes and heterozygotes, and then you have to run the it's sensitive in the PCR conditions. So all those things are there. Application genetic mapping. Second one is your F1 identification. If you have two parental plants and then you have first generation plants, if you run the genomic DNA and then you use the one particular primer for all three plants, they will not have the same kind of a fragments produce amplification. There will be differences. The differences is the variations between the parent and uh, the F1 generation, and what is the dissimilarity between the parents all will be understood in this F1 generation. You can design multiple primers using this technique, you know. You will be able to understand about the primers using this technique. You can select the certain variety. It is used in the plant breeding techniques and all those things. The finally we have AFLP technique, amplified fragment length polymorphism. This is just imagine you have a restriction enzyme digested DNA. Instead of running the agarose gel electrophoresis and southern blotting, you're using this restricted digested DNAs to the PCR. So that's what exactly amplified fragment length polymorphism does. So particularly you are first using a particular DNA enzyme to cut the DNA into the multiple fragments. Now let's say you are having three different strain, you are using the uh, enzyme and cutting the DNA into the multiple fragments, and you will be having the sticky ends. You know what is a sticky ends? Like when you cut the palindromic sequences, you have a product which has a sticky ends. Now, once the sticky ends come, you can see here, even first your DNA, you are having a sequence, and for that sequence, you are using the restriction enzyme. Those restriction enzymes were cut by the uh, restriction sites are cut by the restriction enzymes, and then you are adding something for that DNA adapters. So what is an adapter? Adapter is nothing but it is a double stranded DNA with a overhang. You can see here the red color and it has something overhangs. Now this is able to bind to the overhangs of your restriction fragments. We call it as the adapters. Okay. So basically, it is a certain double stranded with a single stranded overhang, which is complementarily able to bind to the sticky ends of the restriction fragment. So we call it as an adaptase. Once this is bound, we are using has to be done with the DNA ligase so that there will be proper phosphodiester bond is formed between the sequences. Now, then you are using after using you are having a proper double stranded DNA with no overhang. Once you are using adapters, then you are using the amplification with the PCR. So once you are amplifying with the PCR, now again here it will be very easier to design the forward primer and reverse primer. Can you just see here 5 prime end in the both the sides here? Why? Because you know which is your overhangs, you know what is your adapter sequence. You can simply easily design the primers in the adapters. You did not worry about the primer designing. It is very easy to design. So this is about the uh, AFLP. So first your total DNA is digested with the restriction enzymes, normally a frequent or rare cutter. Then you are using adaptation adapters, which will be able to bind with the ligation for these fragments, and then you are using this fragments for the PCR. Now you are having a prime which is complementary to the adapters, and then you are running the PCR. So which is quite easier, same technique which is I have told. So what is the AFLP? Among three techniques, RFLP, RAPD and AFLP, AFLP is very sensitive and it has a wide scale applicability. It is able to understand difference between a heterozygote and homozygote and used for mapping. This is more gives a clarity than the RAPD and RFLP. Highly expensive and requires more DNA unlike your RAPD technique, and then it requires experience for the sequencing the gels. So what is the application of AFLP? Fingerprinting, uh, then mapping, region specific marker saturation, to understand the different varieties, to identify the F1 identification, genetic mapping, plant breeding, and then gene tagging. All those things are utilized in the AFLP. What is the comparison between RFLP, RAPD and AFLP? First, the DNA required for the three techniques. The RAPD requires a very low amount of a DNA, followed by AFLP and RFLP. RFLP requires a large amount of a DNA. The DNA quality. The reason is very simple, because if you have a very less amount of a DNA here in the RAPD and AFLP, after that you are going to use your PCR reaction, you're amplifying your DNA. So here, basically, amount of DNA required becomes less. But exactly RFLP, you're not using the PCR, so there is no amplification. So initially only you require a large amount of a DNA sequence. So you need a more amount of a DNA for this. The second is DNA quality. required for the amplification is very high in the RFLP and RAPD, whereas the AFLP less amount of it. The RFLP is non-PCR based, where exactly RAPD and AFLP is a PCR based technique. The RFLP is not easy, very exactly easy to understand. RAPD and AFLP reproducibility. That means you have to get the same amount, like same specific answers every time you perform it experiment. So that is very high in AFLP and RFLP. RAPD is unreliable. You know the reason, because the sequence is not understood well. The primers are not understood well. It is very random, no prior information. So it becomes very unreliable RAPD. Development cost is moderate in the AFLP, very exactly a bit high. Where exactly RFLP, RAPD becomes easier because AFLP requires the adaptation, adapters, ligation, and then restriction enzymes, where exactly RAPD just requires the primers or random primers, and RFLP requires just an enzymes. Okay, cost for analysis, yes, RFLP will be high and the RAPD, EFLP will be low and moderate. Yes, thank you.